Bone marrow transplantation is performed for different types of leukemias, and it's known that natural killer cells are actually quite potent in terms of controlling, in particular, acute myelogenous leukemia. The genetics of the natural killer system are the main drivers and controllers of the NK function. Therefore, we can look at the genes that transcribe the different receptors that are important for interacting with ligands, and these particular genes will then be predictive of how the NK cell will function in the patient. A large focus of our work has been looking at the KIR family of receptors, which are proteins that interact with the HLA proteins. The gene that we focused on is one of the most ubiquitous inhibitory KIR genes, and this is KIR3DL1. This KIR gene has several alleles, numbering well over 70 at this point. And these alleles interact again with different inhibitory responses upon recognition of their HLA-BW4 ligand. The HLA-BW4 ligand also has multiple alleles, and depending on how you stratify the combinations of these different alleles, you can then develop an operative system for predicting how the NK cells will behave when they're in the patient. What we recognized was that those HLA and KIR3DL1 genotypes that were predictive of high inhibition were actually associated with the highest amount of relapse in our AML patients who received HLA identical transplantation. On the flip side of it was that those HLA-KIR allele combinations that were predictive of a low inhibitory capacity were actually associated with the lowest amount of relapse in our AML patients after transplantation. So what this means is that we can predict, based on the donor HLA and KIR genotypes plus the patient's HLA genotypes, how the NK cells will behave if we do the KIR genotyping to an allele-specific resolution. The significance of these findings is the following. Number one, this is really the first study to demonstrate that KIR allele typing has even more of an effect on transplant outcome than just plain KIR genotyping. The second is that because KIR3DL1 is so common, and because its ligand HLA-BW4 is also so common in our patient population, we can therefore select our donors based on their KIR alleles. Based on this work, we think that donor selection is one that will include not only HLA typing, not only KIR gene typing, but KIR allele typing. And with this, we hope that we will be able to give our patients the best protection from AML relapse.